This is Dr. Bailey, and in this video, we're going to focus on tackling some of the multiple choice for topic 1.1. Now, all the questions that you will find here are questions from past previous IB exams. So there's no questions made up. These are all coming from IB. That means we're looking at IB language and how to tackle it. Now, a lot of students have a hard time understanding the IB questions. They're really good at understanding the information, they study information, and then they come to the test and they're like, what the heck is this guy asking? So what I'm going to try to do is do a couple multiple choice here in this video to help you tackle these multiple choice and to understand more the language of IB. Hopefully this will give you a little bit more strength to tackle the multiple choice questions once you get to your exams. But remember, this multiple choice is just related to topic 1.1, and you will have other videos for 1.2, 1.3, so you get questions more related to the subject of the moment. So with that, let's go. So the first thing in tackling multiple choice is read the question twice. That means the first time you're just reading to know, the second time you're starting to dissect the question. After you have read the question, the next step is to read the four options. Do not make a quick answer by just reading the first and second, because sometimes you will have two options which are there, they are very close to each other, and IB is very picky. They can be very specific, and you end up falling on the trick of answering a question that is right, but not necessarily specific, because, for example, let's just say A and B both are true, but B is more specific, and that's the right answer. IB always wants a more specific answer. Another way also to tackle is after you're reading those four options, start canceling the options that you know for sure are not the answers. That reduces the chances of you getting it wrong. And last but not least, whenever you answer a question and then you start hesitating again, do not change it. So for instance, if you answer B and you go back, you go through the rest of the test, and then when you come back and you think it's A, do not switch to A unless you are 100% sure that it's A. If you still think, I don't know, it might be A, it might be B, always go with the first answer, because that's your intuition, that's your conscience saying that that's the right answer. Okay? So now that you have a little bit of an insight of things, let's start going through some of these questions. We have about 13 questions on topic 1.1, and I'm going to go one by one and let you see how I think through this process. So the first thing we do is make sure you have a pencil or a highlighter in your task, because underlining questions or highlighting questions can help you focus more and trigger the most important things. So first thing we're going to do is actually read the question. It says here, which feature of striated muscle cells allows them to be considered as a possible exception to the cell theory? So that was your first time, get out of the anxiety. And you know that this has to do something with the cell theory and exception. So now your mind is popping with all the possible ideas about this topic. Then the second time, you can underline or you can highlight. Which feature? Now the word feature here, for those who have English as a second language, this actually means trait. So which trait, of striated muscle cells? And when you see the word striated muscle cells, automatically you should go back to your memory that you learned there was 3 examples that were exceptions to the cell theory, and one of them was striated muscles. So now you already started digging. So it says which feature, and you can say which trait of striated muscle cells allows them to be considered as a possible exception to cell theory. So basically this question is asking you, what is it about striated muscle that makes them an exception to the cell theory? This is where you got to go back to your memories of all your studies about the exceptions. Just to be clear, usually we have in the cell theory, we say that one cell is functioning on its own and it has its own cytoplasm, its own nucleus right here, right? And it's doing its work. And then you have another cell with its own nucleus and it will do its work. And it keeps going. However, when it comes to muscles don't work that way. What muscles actually do is that they actually connect all their cells together. So what it would look like is that they will connect all the cytoplasm and they work as one. And this wouldn't be just 2. It will be like a long line of a bunch of cells coming together. And that means now it's one huge cell with multi-nuclei. When I say multi, it means many. So it's one cell with a lot of nuclei altogether. This is what a muscle looks like because they have to function, they have to contract all together as one, and that's what makes it more efficient for movement. So this is what the exception of the muscle cell, the striated muscle cell. It's just they are all bunch of cells together instead of being autonomous. And what does autonomous means? Autonomous means I work independently. It's like saying that this cell here, let's put another drawing here, basically saying this cell here, it works on its own, and this cell here, it works on its own, and this cell works here on its own. That's what cell theory is saying. It's that each cell works functionally on its own. But it's not the truth. They actually work together as one. So it's many cells working as one. So it's not autonomous. So now that we have a little refresh of what the exception of the muscle, now we got to look at the 4 answers. Read them. Don't jump to conclusion quickly. The first one says they are found in multicellular organism. They contain more than one nucleus. You can see that this is kind of looks like the answer. They are specialized in movement, and they're not carried out in mitosis. So you have read them, kind of have an idea. Now remember, try to look for the first 2 sentences or the first 2 options that you can cancel, not the right answer. Do the opposite way. So when I'm looking at this, let's work with the first question. It says they are found in multicellular organism. Now, the question is saying, what is the trait, what is the characteristic or the feature of the striated muscle that makes them an exception? If you say, oh, the muscle is an exception because it's found in multicellular organism. Multicellular means many cells. So we humans, we are made of trillion cells, so we are multicellular. Are you telling me that a muscle is an exception to cell theory because it's found in a body of a bunch of cells? Well, if the muscle is like this, then the heart is too, the lung cells is too, the bone cell is too. No, that makes no sense. All of the cells are inside of us. So that's your first one to reject. You're already going to reject A because it makes no sense. The other one that I will look is in the bottom one. They do not carry out mitosis. Dude, if you do not carry out mitosis, my question is this. Let's put it here. You have an egg, right? And you have a sperm. And when they come together, voila, you have a baby. Now, that baby is made of one cell. How the hell you go from one cell to become a human, right, with so many cells? You need mitosis. And also, how do you want to make muscles bigger? You need mitosis. So D, there's no such thing as it does not carry mitosis. You want to make bigger muscles, you need to make more cells. So you definitely need to go through cell division to make more cells. It makes no sense. It's already canceled too. So then some students might say, oh, they're specialized in movement. Putty, there's no such thing as a cell frozen like this and that doesn't move. Even in your body, every single cell is moving. Blood cell is moving. Everything is moving. It's basically a fluid of your organ. Otherwise, you would have been a rock, like a wall, where every single cell is stuck at the exact position and doesn't move at all. We do move a little bit. It doesn't have to move all the way across the street, no, but they're in a flow motion. So if you're telling me the muscle, the reason why it's an exception is because it moves, I'm sorry to say, all the cells usually move, so that's not an exception. And eventually, it comes to the right answer. They contain more than one nucleus, and this is what I've been explained. So yes, the answer is B. I know that I took a little bit more time in answering this question, but in the beginning, when you're trying to learn this stuff, it is okay to take time with each multiple question because you're starting to learn the language of IB. But with time, when you get to the real test, you kind of going to be a little bit more faster in the logic of thinking. So instead of me taking here, let's say 5 minutes on explaining this, you will take less than 40 seconds. Let's go to the next question. So here says, what evidence from the image of Paramecium indicates whether the organism is prokaryotic or eukaryotic? So that was the first time reading. Second one, what evidence? Now we're highlighting what evidence. That means there must be something that proves from the Paramecium, which by the way, Paramecium is one of the things that you have to study. It's not out of the blue. You have to study as a part of the content. And if you remember Paramecium, it usually is a unicellular. It is unicellular. And unicellular can be eukaryotic or prokaryotic. And this unicellular, of course, it has the cilius for movement right here, right? And it has its nucleus right here. And it has all the other organelles here. Basically, in this one cell, it does all the functions: excretion, metabolism, reproduction, and all those other things. So we kind of had to refresh our Paramecium. Continue with the question. It says, what evidence from the image of Paramecium indicates whether this organism is prokaryotic or eukaryotic? So the question is asking, is Paramecium a pro or an eukaryotic? Now, if I go back to what I just explained here, it has a nucleus. So if it has a nucleus, we know that pro has no nucleus, right? It has no nucleus. Prokaryotics don't have a nucleus. If that is true, we already can cancel B, right? Because prokaryotics is not it. So these two is gone. So now we only have A and C. Those are the options. Now, if you notice, I did not read all of it, right? All the 4 questions, but it's saying that it's pro or eukaryotic. So obviously you got to cancel automatically those 2. So now you're left with 2. Read them. The first one says compartments in the cells indicate that it is an eukaryotic. And then C says the lack of cell wall indicates that the cell is eukaryotic. If you remember from prokaryotics, right? Prokaryotics can be plant and prokaryotics can be animal. Right? And we know the plants, what do they have? Cell wall, right? They have the cell, wall. So plant is an eukaryotic that has wall. And here it says lack of cell wall indicates that they are prokaryotics. The plant here just proved that this sentence makes no sense because plant cells have cell walls and they are eukaryotic. So without even knowing what the word compartment is, you already know that the answer is A. But just to reassure with the sentence, the word compartment here, this sentence here, compartment means that you are found within the wall. So for example, imagine this is a mitochondria. Mitochondria has a wall, it's making a compartment on its own. Or let's say you have a Golgi right here, right? Golgi has a wall, right? And this wall is in a compartment. So they're basically inside of different walls, and inside those walls are doing their own functions. That's what compartment means. Or another word for compartments, in this case in IB, will mean organelles. Okay, organelles or lysosome, ribosome, Brachiar, that's what compartments mean. So a little bit of language here, IB, make sure they know what compartment means. It's basically the organelles inside the cell. So again, the answer is A. Let's move to the next question. Here is a picture of a Paramecium, right? And you can see that this right here is the cilia, which basically is the movement, right? And the nucleus. Always the nucleus would be the biggest circle, and you can see that it's dark. It's a little bit darker than the rest. It's big and dark. Let me erase this here so you can see again the colors, you see? So we know for sure that this is the nucleus. What am I doing? I'm already analyzing before even reading the question because sometimes that makes it easier to go. Now here, you also can have more like a vacuole, right? And this is where you have the storage, you can do metabolism, it can be homeostasis, it can be so many different things. You also have, if you notice here, let me erase this just for an second. You can see that there is foods here, and these foods, what they're doing, they're going inside through what? Through endocytosis. So it is engulfing it. And what is engulfing it? Basically is engulfing food. So that means this is becoming a vesicle and these are vesicles. And what are these vesicles doing? These vesicles are going for digestion. So that means you're eating food. So all these little circles that you're forming are basically digestion. So with that in mind, you kind of already analyze the whole thing. So now let's look at the question. The question says, which function, and now we're talking about function, accomplished by structures X, which is this, so it's talking about this, and Y, which is this one, this is talking about this, in paramecium, which is the picture. So when we look at this, some students say, oh, it's easy. It's basically saying that whatever the X is, the name for it, and Y, what is it is. So for example, if A is the answer, then X should be doing digestion and Y should be doing homeostasis, then that would be answer. Or maybe it's B, where X is feeding and Y is is metabolism. So that's how you read this. This is how you answer a question, because some people have a hard time understanding what this means. So let's look at the options. You're saying the X is digestion, Y is homeostasis. X can be feeding while Y is metabolism. You can also have X is food storage or Y is movement, or X is DNA replication and Y is respiration. Now, how would I be tackling this? I have to go back and look at here. Now, I can start by canceling the D. Why? Because it says X has to do with DNA replication. We know that DNA replication happens in the nucleus. So if X was basically in the nucleus, the line should be going all the way to here, and it's not. So automatically this is canceled. The next, let's see, food storage. C is food storage and Y is movement. So here is where we can say the X is saying the X is food storage. Now, I'm going to erase this to be a little bit more clear so you can see what I'm talking about, right? So we can say X is food storage, and you'll be like, oh, maybe it is food storage because look, it's like food is coming in, right? It's coming in and it becomes storage. Okay, I would say if this is your logic, fine. But then when you look at Y, it says that it's movement. If it's movement, it has to be outside of the cell because it's the outside that moves the thing. But the Y is nailing inside. So even if you don't know about the drawing, you can see that it's not a movement. So C is out of the way. So now we're left with A and B. Since we're already left with A and B, let's do this now. What is A and B? Let's see. Now, if you look at X, right? X, you're stuck between digestion and feeding. Okay? You'd be like, oh, it could be feeding because it's the food came in, but it can also be digesting because the food came in and then this thing is just breaking its points, right? It's eating it. So the X is kind of giving you a little bit of doubt. Now you look at the Y. Y basically is this thing right here. It says it can be homeostasis or it can be metabolism. But the metabolism is the same thing as feeding. It's breaking down food. So it does not make sense that this is breaking food. This is more for metabolism. This is more for homeostasis. This is like a vacuole, right? It's a vacuole that takes care of storages and work with things. So feeding and metabolism is the same thing. So that's canceled. So in the end, you only have the option is A. So the Y is for homeostasis and the X is for digestion. That's the right answer for this one. Next, what feature of both striated muscles and aseptate fungal hyphae is different from a typical cell structure? So reading the question again, we're talking about feature. Again, what is the trait? And now we have two exceptions, the striated muscles and the aseptate fungal hyphae. What makes them different from a normal cell? What is it about them? Now, a normal cell, usually it will be just a nice little circle with the nucleus inside. We already know that striated muscle it's a bunch of cells that came together as one, so now they are made of multinuclei. That's what a muscle is. Now, what about the hyphae? Hyphae is usually found under the tree where there's the fungus, right? And then you have these nice long root hairs coming down, and these are hyphae. Now, if you zoom in and you look in, it looks like a long cell. And again, its cytoplasms are connected. So with that, it is the same thing as the muscle. So they both having a lot of nuclei in there. So let's look at the four options. They have multinuclei within a structural units. Now, just by reading the first question says, oh, it must be that one. But do not do this. Make sure you read all of them. The second one says they have a cell wall that is not made of cellulose. They have a plasmid. And they have the absence of cholesterol in the plasma membrane. So go with the counseling. Let's look at the first one. They have a cell wall that is not made of cellulose. Now we know that animals do not have cell wall. Humans do not have cell wall. So if it's a muscle right here that is coming this, how is it that a muscle have a cell wall? They don't. So this is already discarded. Next, plasmid. Plasmid is for bacteria. And muscle is not part of a bacteria. So automatically, this one is done. It's out of here. Because remember, we're talking about a feature of muscles and hyphae. And these guys are not bacteria. So it's canceled. Then they have an absence of cholesterol in the plasma membrane. Now, here's the thing. Every single plasma membrane in the body needs cholesterol to keep it fluid. So if you don't have cholesterol in your plasma membrane, you're most likely that your membrane is going to be hard and you're dead. So it can't be this one. So you go back to A. Now you see they have multiple nuclei, which is true. They both have multiple nuclei in that one structure unit. So yes, the answer is A. So this is a good thing. And again, I know that I took it longer by canceling these sentences, but again, always cancel the sentences is very important because it makes sure that you are not choosing an answer that might be right, but there might be another one that is more specific. Next question. When compared to other body cells, which characteristics of stem cells is the most important for therapeutic use? Now this, you have to know the information about stem cells. So let's read again. So we're comparing to other bodies, other types of cells in their body. And what is the trait? What is the characteristics of stem cells? What is it about stem cells that makes them so important for therapeutic use? So we know that stem cells, they usually are not, they don't have a function yet. They're just a cell. So they're brand new, if you want to keep it that way. And when I say brand new, it's the same thing as saying that they are not differentiated. What it means by this? Not differentiation means they don't have a specific job yet. So this is a stem cell, and then eventually it can become a heart cell, right? Or this stem cell can become a lung cell, or it can be a muscle. Now, these guys, the lung cell, the heart cell, the muscle cell, these guys have been differentiated. That means they have now got a specific function. So you cannot turn a lung cell into a heart cell. And you cannot use the lung cell to fix a heart. You cannot use a heart cell to fix a lung cell. You cannot use a muscle cell to fix, I don't know, let's say a liver. You can't, because they already have specific functions. They are not stem cells in this case. So what is amazing about the stem cell here is that it still hasn't got a specific function. He has not been differentiated. So then let's look here at the possible options. First, we say less differentiation. This sounds like an answer. Less excretion. The word excretion means pooping, peeing, getting out of your body. C, lower rate of reproduction. Okay, we know that stem cells can reproduce, right? They can make self-renew. That means they can make copy of themselves, and then they can make extra copy of themselves through mitosis. That's one of the biggest traits about stem cells, as they can replicate themselves. And of course, the last thing is the lower rate of metabolism. There's no such thing as one stem cell that has a high metabolism while the other stem cell doesn't. Like, all the stem cells are the same at this point. So now that I've gone through it, let's cancel. We know that there's no such thing as a lower rate of metabolism. All stem cells have exactly the same rate of metabolism. Low rate of reproduction. No, stem cells, you can self-replicate. That means you can do mitosis as many times as you want. Less excretion. You're telling me that a stem cell cannot poop out. But wait, if it doesn't poop out, that's a problem for the stem cell because there's wastes. So it needs to be getting rid of it. Now, this is more likely, less differentiation. That means the stem cells, they haven't been differentiated. Stem cells can become any kind of cell. They're not like the lung cells, the heart, or the muscles. These guys, they already have been differentiated. That means they already have given a job. So the answer here is A. Next, if the cell of a multicellular organism have the same genes, how can there be many different cell types in the body? Now, let's read this again. It says, how cells of a multicellular organism, in this case, it can be, let's say, human, because human is made of a bunch of cells, right? How does cells of multicellular organisms have same genes? So basically, this question is saying that these are a bunch of cells in your body, right? And all of these cells have the nucleus, right? And they're basically saying that all of these nucleuses here, they all have the same genes. So let's say there's 50 genes on this one. There'll be 50 on this, there will be 50. And all of these 50 is exactly the same. So it's 50 identical genes, and all of these 50 genes are found in every single one of these cells. Now, because all of these guys have the same genes, right? They all have the same genes. The question here says, how can there be many different cell types? So imagine if they all have the same genes, however, this one came out to be a heart, and this one came out to be a lung, and maybe this one came out to be a leg. You're like, wait, if all of them have exactly the same genes, but one cell came out to be a heart and another cell became a lung, how does this work? How is it that each one went to have something different? And here is the rules. Let's erase this so I can give you space. So imagine all of the cells are just like this one right here, right? And in the cell, I'm going to put here the genes. Let's say 1, 2, 3, 4, 5. Let's say each one of these is a gene. Now, every cell, let's say there's 100 cells, and all of them has exactly these 5 genes, right? The difference is where you're located. So, for example, let's say you have a box here, and you have cells on the outside. If these cells are located on the outside, on the corner of the box, then gene number 1 will be expressed, and you will make a specific protein. Okay? Remember, all of these guys have these 5 genes, exact same thing. Now, if you are in the middle of the box, like kind of in the middle, not the core, but the middle, then if you are these ones, then maybe this is the gene that will express and produce another type of protein or another type of function. And then you have other cells that are found in the middle. If you're in the middle, then maybe you express these 2. And this will give you another type of function, another type of protein. So look, all of these cells here, right? All of these cells in this box, they all have those 5 genes, like exactly the same genes. The only difference is that in which of these cells, depending on their location, right? If you are on the outside, you are expressing this. If you are on the inside, you're expressing this. And if you're in the middle, you're expressing this. So it's about the location. Okay, so depending on the location, you will have some of the genes expressed, while in other locations, you will have other types of genes expressed. That's what it means. So with that in mind, let's look at the options. The first one says some genes, but not others, are expressed in each cell type. This looks very close to be an answer. Second one, the cells lose some genes as development occurs. That doesn't look like it. We do not lose our genes. Our genes will stay with us until we die. Next, genes do not determine the structure of the cells. Then why the hell do we have genes, my friend? The genes are found in the nucleus. They are the information. They tell you how to do things. Makes no sense. D, cells must practice division of labor in order to survive. The cell does not practice division of labor. Like, that doesn't mean anything. The cells, they function as a unit. They do all of it together as one. So they do not divide labor. This has nothing to do with the expression of genes. So let's cancel. I know for sure B, lose some genes. We already know that they don't lose genes, so that's canceled. We also know that it says here that genes do not determine the structure of the cell. It does, they do determine. So these 2 are gone. So then you'll be stuck between some genes but not others expressed. That looks more likely. And this one, they must practice division of labor in order to survive. But What does this has to do with how you can make different cells have different functions, right? They have the same gene, but each one is different. And this is saying, oh, because they have to have different jobs. Well, you might have many heart cells and they're all doing the same job. When you talk like this, you want to make sure they have different labors, then there should be just one cell for your heart, one for your lung. That would be really funny. Imagine you have one heart cell, just one lung cell, and then just one leg cell, and then one finger cell. You probably will be made of maybe 10 cells maximum in your life, and we are made of trillions of cells, so nah. Now, some genes, but not others, are expressed in itself. It makes sense. So that's just like the drawing that I did there, right? You had inside here, you had these genes, depending on the location. If you are on the outside, you are expressing this one. And if you are more in the inside, then you probably are expressing these. And then if you are deep in, then you most likely are expressing this one. So you see some genes are expressed while others not. So the answer is this one. So the answer is A. Let's move on to the next one. Which method provides the biochemical evidence that cells have begun to differentiate? So first we need to highlight here which method. So it's a method and it's providing what? Biochemical evidence. So what is biochemical evidence? Biochemical evidence in this case is talking about structures. Like for example, here's structure. This is a structure of biochemical C, or you can have an amino acid attached to amino acid attached to amino acid. This is a protein, right? Amino acid. Or you can have a ribose here, right? C. I'm not doing the whole drawing, but you can get idea. It's chemistry, right? Biochemical, it's chemistry in the real life of living things. So we are looking at these guys in this level, okay? Now, which method provides biochemical evidence that the cells have begun to differentiate? So we're trying to say that somehow the cells have begun to get their job, right? Because the word differentiate, if you remember from stem cells, differentiate means I'm having a job, like a cell having the job to pump, so it becomes a heart cell. So let's look at the options. Now, before I get to the options, just make it clear, right, that we're talking about what is the method that provides biochemical evidence. So if you observe in a light microscope, right, you're observing in the light microscope, can you actually see biochemical evidence? No, because the microscope, the maximum you can see on those nice little circle thing is cells like this, right? You see the entire cell. You cannot see the details like this, like the structures and chemical. Then this will be canceled out for sure. There's no way. Observation of the electron microscope. Well, we could see electron microscope, but the problem with electron microscope is that it's dead. What I mean by that is that means inside the microscope, you can only see things that are dead. So how can you see cells beginning to differentiate? If they're beginning to differentiate, that means they're alive. So you're watching them become differentiated. And in an electron microscope, you only see dead cells, so you can't see them differentiate. So this is canceled too, right? Next, X. So then examination of DNA sequence. Well, okay, it could be DNA sequence, but what it means examination of DNA sequence, it means A, T, C, C, G, G, G. That's what you're doing. You're checking the sequence. Or maybe the other one is A, T, C, G, 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 G. So you're examining the sequence that is going on. Now, can this tell you if it has been differentiated or not? Not really. Why? Because again, I'm going to show you this through the drawings that I did before. You can have the same gene. And when I mean the same gene, as I mentioned before, all the cells have the same gene, which that means they have the same sequence of A, T, whatever CGs, same ones. That's basically what it is. So examining it is just looking at these letters. Will this tell you if he has differentiated or not? Not really. It will not be a proof of anything, because all the cells, let's say you have 100 million cells, right? All those million cells will have exactly the same sequence here, the same here, the same. So examining, looking at them is not going to make you see them differentiate into a heart, into a lung. So that leads me to the only question, which is the analysis of proteins. Now, proteins, they are biochemical, right? And what I mean by biochemical is they are like this: amino acid attached to amino acid. And each of these proteins have a specific function. So the proteins that you find in your heart are different than the proteins you find in the muscle. And whatever the proteins that are working on your heart, they usually are for pump. The proteins that are found in the muscle is for movement. The proteins that are found in the intestine is for digestion. So by analyzing these proteins, what are you doing? You figure out what kind of job they're doing. Because again, the word differentiation means job. And each job uses different types of proteins. So the right answer would be the analysis of the proteins. See, by you noticing what is the function of the protein, you can know what kind of differentiation or what kind of job it did. And again, protein is a biochemical evidence. That means you're going all the way to the structure. So the answer in this case scenario will be D. All right, this one is where students see this and they start to freak out. They're like, how the hell do I do this? First, you have to remember Louis Pasteur's famous experiment, which they were trying to prove that cells, right? Cells come from previous cells. So let's say this is an old cell, and you can only create a new cell if you had an old one before it that was actually living. That's what Pasteur experiment is about. But before Pasteur experiment, they used to have this idea called the spontaneous generation, which is right here, the word spontaneous generation. What does spontaneous generation mean? It just means that out of thin air, boop, a cell just show up out of nothing. It was like magic. It just poof, appeared out of nowhere. So they think that cells come from, what did the cell come from? This cell came from nothing. There was nothing before it. It just popped out of the blue. That's what spontaneous generation means. And what Pasteur was trying to do is, this guy right here, he was trying to say, hey, there's no way that a cell just appeared out of magic. That cell, if this cell is alive, it's probably because that cell came from a previous cell, right? And this cell here, previous one, should have been living in order to make this new one. And Pasteur tried to prove through this experiment that the spontaneous generation, this theory that cells just pop out of nowhere is the truth. It's not really. So this experiment here is talking about Pasteur proving that spontaneous generation is wrong. Now let's look a little bit more focus. So here we have 4 examples of flask, right? Because he was working with broth. Broth is like a soup, right? And he was putting in flask. Now there's 4 scenarios. Let's look at each scenario. So in the first scenario here, it says not boiled, okay, and not sealed. If it's not boiled or sealed, if you have bacteria, let's put in another color of orange here. You can have bacteria here living. And if you have these bacteria living, this means that these bacteria just pop out of nowhere, right? You didn't boil, you didn't burn them, and it's not sealed, so anything can just come in. Basically, you have kept it open, but at the same time, before you even boil this, before you put this thing here, before you actually put this little flask here, and when you put the broth here, you already had bacteria on it, okay? On this one, it was boiled. So that means you put fire here, and that means you actually cleaned all those bacteria. That means these guys was cleared. That's why you see it's cleared. But look at it's not sealed, so it hasn't open, it's open. What is more likely, if it's open, then you have these little bacteria on the air. They can just come in here and then land and start making new ones. Then you have the third one. This one has been boiled and then it was sealed. So sealed, the bacteria will try to come in, but they'll be stuck here. They cannot even go much more. So here it's empty. There is nothing. Here is just soup, no bacteria. In this case scenario, it was also boiled. So that means you sterilize the whole thing, but it was not sealed. But the bacteria will try to come in, right? And when they come in, they're gonna probably be stuck here. The bacteria will not move in, so what happens? It continue being clear. That means the chicken broth has no microbes, no infections. However, so looking at the four scenarios, these two, you will have microbes in here, and the last two you don't. So now that we have looked a little bit, let's look at the options. It says here, what results would expect with no spontaneous generation of life? Now, students will say, what do you mean no spontaneous? That means in which these guys would you expect to not have a spontaneous generation of life? That means it doesn't show up out of nowhere. So we have decomposition of broth by microbes occurred only in flex W. So basically saying, when we say decomposition of broth, we're talking about having the microbes. Where is it? They probably have written somewhere the microbes here, have they? Oh, right here, see? The microbes, what do? They're eating the broth, so they're decomposing the broth. So they're basically saying that the composition of broth by microbes occurred in flask W. Was there microbes here? Yes, there was. Was it actually breaking down? Yes, it was. The next one says, decomposition of broth by microbes occurred only in flask W and X. There was decomposition here because microbes were here, and there was also decomposition here because there's microbes here. Okay, so far B looks good. Now, C, decomposition of broth of microbes occurred in W, X, and Y. So it did work in W, did work in X, and Y, we don't see any dots orange here. So there was no microbe inside, so it did not digest the broth. So C will be out of the way. At last, decomposition of broth microbes occurred in all of them. Well, Z also, you don't see any dots of orange here, which means there was no microbe. So in these two case scenarios, there is no microbes eating. That means microbes didn't appear in this way. So the right answer here is B, because again, B shows that both the W and the X, they both have decomposition broth by microbes, right? They both have the broth being broken down by the microbes. So the answer being at B, this results will be expected with no spontaneous duration of life. They were expecting it, but you can see it was broken down. So that's for B. Let's go for the next one. Now this is an electron micrograph. Shows a thin section of a plant mesophyll. This has to do with magnification. If you remember the equation, magnification, which is M is equal to the image over the actual size. So the image here, this one is actually the drawing that you see on the microscope. And then this right here, right? This is the actual image of the whatever microbe or whatever the cell is. So this is in real life, and this is the one that we actually cannot see. Magnification, it means how many times bigger. Bigger it is. Done in a microscope. So we have that. So we have all this done. Now we have to go through the question. The question says, what is the magnification of the image? And then the students start freaking out. Wait, we don't have this, and we don't have that, and we don't have this. And I don't know, there's only a picture and we have this little thing going on, and they freak out all over the place. Trust me, all the answers you want is in the picture. But the first thing you need is a ruler. Always take a ruler with you to the test. The second thing is this scalar here is what gives away what will help you answer these questions. So let's tackle this. Make sure always have a ruler to go through this. Let me erase this to give you some space here to draw this. All right, so how would you work with this? First, you get yourself a ruler, and you have that ruler right here. And this ruler is you getting the size of this guy. So you getting here a ruler and you're measuring the guy, right? And let's say it came out, in this case, it came out 30 centimeters. Actually, let's say 3 centimeters because this drawing will make more sense. Oops. So let's say it's 3 centimeters, okay? So being 3 centimeters, now that's what? That is actually the image because you are measuring the drawing. So measuring the drawing, not the real size. Can you find the actual size? Yes, you can. How does it work? This is where that scale that I showed here comes to effect. So you see that how many times can you put the scale? Probably 2 times, right? So that means it must be 10 micrometers. So the actual size of the specimen is 10 micrometers. So now, as you can see, I have solved 2 things. I have found the actual size and the image, knowing that the equation is image over the actual size. Just to recap, how did I find the image? I just took a ruler and measured here with the ruler and it came out 3 centimeters. Then, how did I find the actual size here? How did I find this guy? Well, I took the scale here and I measured how many times can I repeat the scale and came out twice. So that's the image drawing and this is the actual size. And I want to find the magnification. So when I put in the magnification, I will have 3 centimeters over 10 micrometers. And this is the next mistakes that students do. The biggest mistakes they do with this is that they forget that these guys cannot be in different units. They have to be in the same unit. So you can change. You can go put micrometers to centimeters, or you can take the centimeters to micrometers. I usually would go from the centimeters to micrometers. Now, for students to understand a little bit more about this, what I would do is use the middle school way of getting things done. If you don't remember, some people automatically know the answers. They just know that this has to do with actually putting 4 more zeros up there. But if you don't, write meter, decimeter, centimeter, millimeter, micrometer, and then nanometer. But we don't need the nano for now. So if you have 1 meter, it's 10 decimeters, it's 100 centimeters, it's 1000 millimeters, and then for micrometers, you have put 3 zeros. Remember, it's 3 nanometers would have been 3 zeros also. So if you have 3 centimeters right here, how many would that be? Obviously, you can see 0 plus these 3. So it's 30,000, the answer. So with that in hand, it's 30,000 divided by 10. And that 0 can cancel with this, so it's 3,000 times. And voila, you have the answer. The answer is C. So remember, whenever you have these micrographs, make sure you have a ruler to measure the measurements going out this with the ruler, and then use the scale to measure how many times can you put the scale in the drawing. This will be for the image, and this will be for the actual image, and you can find the magnification. So it's not that hard. Next question. The Stargardt disease, a genetic disorder that causes vision loss, is being treated with using retinal cells derived from human embryonic stem cells. Why are embryonic stem cells used? Like, you notice this, the first sentence here is just giving you so much information, and you start freaking out and Sometimes it's good, but then it gives you the real question. Why are embryonic stem cells used? So sometimes it reads the whole thing, and then the sentence goes says, why are embryonic stem cells used? Now, if you remember, embryonic stem cells they're used to treat diseases, right? Because you can take one little cell, stem cell right here, and you can convert it into a heart cell. You can convert it to a lung cell, or you can convert it to a muscle cell, whatever it is. One sec. Sorry. So in this case, that's what stem cells use. But just to be clear, let's look at the background. We know that Stargardt disease here, this is a problem with vision. You lose your vision. Why? Because the retina in your cells, the part of your cells of your eye cells are being destroyed. But you can use the stem cell and create new retina cells. And if you create new retina cells, you can replace the dead or the destroyed retina cells with the new ones. And voila, the person has the vision back again. So what are the options? They already specialize in this function. So here it says, why are the embryonic stem cells used? This first sentence is saying that stem cells already do the retinal job. That's not true. Stem cells here, they're undifferentiated. That means they don't have a job yet. So I would even cancel this. I wouldn't really waste my time with this. The next thing it says, they're able to differentiate into required type cell. This makes more sense. They retain stem cells properties after specialization. They can retain, but this has nothing to do with helping the vision loss come back. There are no ethical issues concerning. Oh, there is. Because if you take some of the stem cells of a fetus or an embryo baby, you destroy the baby. You cannot do this. If you want to recap here, where'd you get the stem cells? The stem cells, when you have the sperm and you have the egg and they come together, they become a zygote, right? And then this goes through mitosis. And when it goes to mitosis, you start having so many of these cells. All of these cells that I'm drawing right now are stem cells. Now, if you go in the lab and you take one of this out, use this stem cell to make a retina cell, what do you think happens to this baby right here, this potential baby? Well, guess what? It eventually dies. It's destroyed, it's hurt. So there is an ethical issues when you're taking these stem cells from the embryo, because the baby is most likely to be killed, the potential baby. So this one can also already guarantee that it's canceled. So now we have between B and C. Now the first one says they are able to differentiate into required cells. That means they're able to become the job of retina cells. This looks like the prominent answer. C, they retain the stem cells properties. When it says retain, that means they continue to be with no function after specialization. The word specialization means they have been specialized, they have been differentiated. That means now they have a job. Once you get a job, you cannot go back to being stem cells anymore. So that means you cannot retain means hold the stem cells properties. What are the stem cells properties? The fact that they have no job. They have not been differentiated. So once you have specialization, let me erase because there's so much on this now, messy. What do I mean by this? With the word specialization, in this case scenario, it's saying that now you have a specific job. So let's say an example, you have become a liver. So now you have a job. Your job is to do the liver job. Once you do this, you cannot be a stem cell because as a stem cell, you have no specific job. And we say this as known as undifferentiated. While this case, the liver has been differentiated, or you can say it has been specialized. So it can be a liver and have the properties of the stem cell, because the stem cells can become anything else, right? A stem cell can become a heart, a stem cell can become a lung, a stem cell can be a muscle. Well, liver cannot become a heart cell. The liver cannot become a muscle. So you do not retain the properties of a stem cell at all. So C is canceled, which leads me to one and only answer. The only possible answer for this case scenario will be this one. It's able to differentiate into a specific cell type. In this case, it will be able to do what we call the retina cell, right here. It can be able to make this guy. Oops, so again, the answer is B. I wrote it wrong. Next, Which sequence has the cells arranged according to their ability to differentiate, starting from the least able? Again, which sequence, so we're talking about a sequence right here, right? Has the cells arranged according to their ability to differentiate, starting from the least able. Now, let's go back to what differentiate, this word right here, what it means. Differentiate means in the beginning, you have no specific job. And then suddenly you become a specific job, like a heart job, or you become a lung job, or you become a liver job. These have been what? Differentiated. And this one, still no specific job. So they're saying to make a sequence and arrange according to the ability to differentiate. That means starting from the least able. That means the one that cannot differentiate to the one that can differentiate. So if you had to put on a line, how would it be? It would be a line like this, right? So you want to go from here where it's starting from the least able. So this means no, you cannot. You can't differentiate. And here, yes, you can. So let's look at the first, in this case, let's look at the first one option. It says bone marrow, then a neuron, then embryonic, then umbilical. So if you put the bone marrow right here, no, you cannot differentiate. Well, bone marrows do have stem cells, so it can differentiate. So between bone marrow and neurons cannot differentiate. So neurons should have been here instead of the bone marrow. So A is out. Next, neuron. Once you're on neuron, by the way, who don't know what neuron is a nerve cell. So that means you have been fully differentiated. That's your job. Bone marrow, the good thing about bone marrow is that bone marrows produce stem cells, so that means you can differentiate. Umbilical cords, these are still on the baby, so it still has stem cells. It can differentiate. And embryonic, it's actually the baby, so it can definitely do it. So B looks like a promising answer, but still, let's continue. Here, umbilical. Umbilical has stem cells. If you put it up here, it says, no, you cannot differentiate. That's not true. Umbilical has stem cells. It can differentiate. And then it says the least embryonic will be the next one. Neuron, if you put it here, it says you're basically saying that the nerve can differentiate. No, that's not true. C is out of the way. Which that means D is also because, again, it's saying the neuron here. So the only answer you have is B. Because, again, it says to do the sequence, and it tells you from the least able. So you're going from the one that least can differentiate to the one that then you can actually differentiate. So neuron cannot differentiate, but the embryonic can. And then, of course, let's go for the next one here. This is cholera and paramecium, are both unicellular or eukaryotic organism livings, okay, with fresh water. Cholera is photosynthetic. That means it has chloroplast, it makes photosynthesis, and it has a cell wall. Which organelle will be found in paramecium but not in cholera? So when you're thinking about this, we got to think about plant cell and animal cell. So looking at this, we're going to look at the different options. So we have to think about chlora. Chlora is usually right here. It has a cell wall, right, which is the two lines here. And then it has a plasma membrane. And then it has probably the chloroplasts around here. And it has a nucleus, right? So that will be the chlora. Now, they're saying which organelle will be found in paramecium but not at chlora. And in this case, we're looking here. We know from the question, A, chloroplast. Which organelle can be found in paramecium but not in chlora? Well, paramecium is not photosynthetic, right? Which organelle can be found there but not in chlora? We already know that chlora has photosynthesis, right? So chlora has chloroplast, right? Because without chloroplast, you cannot do photosynthesis. So if you're saying that chloroplast is only found in paramecium and not here, that's not true, because obviously here gives away that this has chloroplast, so it's not A. Contractile vacuole. Paramecium usually has a contractile vacuole because this is where it does its work. It has a vacuole, as if you remember the vacuole that it looks like a drawing like this, and this vacuole is making the functions, is doing the homeostasis. Now, do chloro have a contractile vacuole? Even if you don't know, let's continue. Here you start having doubt. This is where students usually have doubt. Then you have the C, which is rough endoplasm reticulum. What is a rough endoplasmic reticulum? This is where it produces most of the proteins, right? Now, do plant cells have rough ER? Do Paramecium, plant cells or animal cells do? So both plant cells, animal cells, they do have rough ER. They do produce proteins, right? Otherwise, how would they go through process of all those things they have to do, such as movements and stuff like that? So they both have to have this. So not right. Next is chlorua and Paramecium. What is it that you can find in Paramecium? You can find mitochondria. Yes, you can, because you need the metabolism to do the things. But guess what? Chlorua is also like a plant cell. It also has a mitochondria. So they both have it. So the only thing we have here is left is the contractile vacuole. The chlorua is much smaller and it doesn't need a contractile vacuole for storage. So the only option you have here is the contractile vacuole. That's what Paramecium has, while chlorua you cannot find it in chlorua. So this you could have done by just guessing, killing other ones. But before I even get to these stuff, if you actually did study the chlorua and Paramecium, you would know that Paramecium has the contractile vacuole. So this is based on knowing the information from your studies. But what I did here was trying to, if let's say you forgot to study, how would you cancel? Once again, cancel the chloroplast because chlorua does have it. Also, since these two, they need to function, they need energy, so it cancels this. And the rough endoplasmic reticulum, they both need protein in order to do their work, right? Why? Because proteins can turn into enzymes, and if you want to do photosynthesis, you need enzymes. So chlorua does need that, so cancels. The only thing left is this. So this is another way of you canceling all the possible answers and going for the educated guess. But if you did do those studies, you would know exactly the contractile vacuole is the only one. And we're getting to the final question of this video. Which living structure is an exception of the cell theory? So we are talking again about a living structure, and we're talking about it does not go with the cell theory. This, after so many times, you kind of get the answers. You automatically, I know that you guys would automatically go to the answer. Oh, it's striatal muscles right here, because we know the striatal muscles is an exception. But please read the rest, just to make sure. A single cell alga carrying out all the functions of life. That's not, because we're saying that one cell function unit does the whole work. So, no. Artificial synthesis of the organic molecules with urea. This is not an exception of the cell theory. This is another type of theory. This is talking about vitalism, so it's not it. A multicellular organism with cells undertaking specialized roles. Every single cell will have specialized roles. So this is an exception. So this is part of the cell theory, basically saying that every cell has its function unit. So obviously you already have the answer from the start, but just going through the sentence to make sure that that's the answer. And these are just examples of tackling multiple choice questions on topic 1.1. I hope this kind of did a little bit of help of how to tackle this. Good luck.